Hey guys, so today we're going to talk chapter 7.1. Chapter 7 is all about membranes and their function with cells. So we like to consider cell membranes the edge of life, and that's because a single cell can live on its own, and all cells are enclosed with a plasma membrane. So the plasma membrane is a boundary that separates out the living things from its surroundings. The plasma membrane is considered selectively permeable, which means that some substances are allowed to cross through, while others may have more difficulty passing through or require energy to pass through. So phospholipids are what is actually the composition of the plasma membrane. Plasma membranes contain both a hydrophobic and a hydrophilic region. Typically, the phosphate head, which I'll show you in a few slides, is your hydrophilic region where it interacts with water. And your hydrophobic region, or your water-fearing region, is the fatty acid tails that stay on the inside of the membrane. The plasma membrane is also said to be a fluid mosaic model. And this model states that a membrane is a fluid structure with a mosaic of various different proteins that are embedded in it. A quick scientific inquiry on how we discovered what the plasma membrane looks like. Membranes have been chemically analyzed for years and were found to be made up of mostly proteins and lipids. Scientists who were studying the plasma membrane reasoned that it must be a bilayer when they did something called a freeze fracture model. So this right here is a typical phospholipid, one of these, where this is your phosphate head, and these are your fatty acid tails. Your heads are hydrophilic because they interact with water. Your tails are hydrophobic because they do not like water. Think like oil and water. When you put oil and water together in a beaker or in a bottle, they'll separate out. And that's because oil is hydrophobic. This all started in 1935 with Hugh Davison and James Donnelly. They proposed a sandwich model in which the phospholipid bilayer lies between two layers of globular proteins. This was later found to be not true because they found proteins in the membrane that had both hydrophilic and hydrophobic regions. In 1972, S.J. Singer and G. Nicholson proposed that the membrane is a mosaic of proteins that are randomly studded throughout the membrane and dispersed throughout the bilayer, and only the hydrophilic regions of both the membrane and the proteins were exposed to water. This is that mosaic model that they were able to come up with, that proteins here that have both hydrophilic region and hydrophobic regions, and the inside studded randomly throughout a plasma membrane. That freeze fracture study of the plasma membrane supported the fluid mosaic model. This is a specialized preparation technique that splits the membrane between the hydrophilic region, so pulling apart those two fatty acid tails to expose half of it so they could study its composition and what was inside of it. So this is what ended up happening. They did the freeze fracture of a cell, pulled off half of that membrane to study what was it like. Membranes are very fluid. Phospholipids in the plasma membrane can move throughout the bilayer. Most of the time they shift side to side or laterally, but on occasion they can flip transversely across the membrane as well. This is that mosaic that I was talking about. So Our cell membrane is considered a fluid mosaic. Fluid because it moves and these phospholipids are not stuck in place. And then a mosaic because our cell membranes are randomly studded and no two are perfectly identical. Some proteins go all the way across, some sit on the inside, some only cross halfway. We also have cholesterol in the membrane and carbohydrate tails here that act as identifying tags. And then some of our proteins hook onto this extracellular matrix. This is that movement. So most of the time, about 10 to the 7th times per second, the membranes are moving laterally across the membrane. And about once a month, membranes will flip across the membrane. As temperatures cool, membranes do switch from a fluid state to a solid state. The temperature at which a membrane solidifies depends on the type of lipids that are inside of that membrane. Membranes can be made up of unsaturated fatty acids, which are more fluid, or it could be rich in saturated fatty acids, which tend to not be as fluid. Membranes must be fluid in order to work properly. Usually they're about as fluid as a salad oil. So they stick together, but they are fluid like a salad dressing. Think of like an Italian dressing or an oil and vinegar style dressing. That's about the fluidity that you're looking at for membranes. Cholesterol is added to the membrane to help strengthen the membrane so it doesn't completely break down. And it has different properties as well. At warmer temperatures, cholesterol restrains the movement of the phospholipids, not allowing it to completely fall apart when it's super warm. But when it's colder, cholesterol helps maintain the fluidity by preventing the tight packing of the phospholipids together. So here are your unsaturated fatty acids. They tend to bend. So there are gaps in the membrane where our saturated fatty acids are very tightly packed together, and then our cholesterol comes in and can fill in some of those gaps to restrict movement, but also to aid in movement depending on the temperature that you're looking at. There is an evolutionary significance to the different types of lipids that are inside the proteins or inside the membrane. Variations in lipid composition of cell membranes appears to be an adaptation to the specific environment. Where we live, we have more of an adaptation to our cell membranes becoming more and less fluid throughout the seasons. But someone that may live in Florida and comes up in the middle of winter feels very cold. They tend to wear extra layers, they drink hotter beverages, they take warmer showers. And that's because being down in Florida, where it is typically 70 degrees or higher year round, they have adapted to that style of lipid in their body. The ability to change the lipid composition in our cell membranes in response to temperature has evolved in organisms that live where temperatures vary. Typically up north, where we are in New England, our temperatures vary all of the time. And that is an adaptation that we have as humans, that it takes us less time to adapt to the colder climates because our body is prepared for it. Membrane proteins have many different functions. They're a collage of different proteins in the membrane that are often grouped together that have one goal to achieve. They can be embedded in the fluid matrix of the bilayer, and they may even float on the surface of the membrane as well. The proteins determine most of the membrane's specific functions as to what it's doing and why it's doing it. Peripheral proteins are those that are found on the surface of the membrane. They do not cross both sides. They may cross one side. You can have an interior or an exterior peripheral protein. Integral proteins penetrate through the hydrophobic core and emerge the other side. Those that span across the membrane are called transmembrane proteins. That hydrophobic region of the integral protein consists of one or more stretches of a nonpolar amino acid chain and is often coiled into an alpha helix to protect it from any water that could potentially get through. So this would be a transmembrane protein with an alpha helix inside. There are 6 major functions of proteins. Transport is its number 1. Each protein will transport a different molecule or macromolecule in the cell. Some take energy, some do not. They also are involved in enzymic activity, signal transduction, cell-to-cell recognition, intercellular joining, and attachment to the cytoskeleton and the extracellular matrix. So here you see transport. Sometimes it's like an open doorway and these ions can just move freely in both directions in and out. Other times it must change shape by the use of energy to allow things to come through or to leave. Proteins can take place using enzyme activities with building different enzymes for the body. Signal transduction. It takes in a signal from another cell and sends that message into the cell. Cell-to-cell recognition. This could be a skin cell coming in contact with a muscle cell that just saying like, hi, I'm a skin cell. Intercellular joining. This happens in cells that need to be tightly packed together. And then we also have that attachment to the cytoskeleton or extracellular matrix. We will pick up with this in our next video.